Good afternoon, good evening. Welcome everyone to the session on innovative clinical trial designs to accelerate increase in PrEP choices. Next slide, please. And again, I welcome everyone, and I wanted, go ahead, next slide, please, to just indicate that this symposium is sponsored by the HIV Forum's project called Innovation in Clinical Trial Design for New PrEP Products. And I want to thank and acknowledge our three project co-chairs that include Helen Rees and Ken Mayer. And I've given here the link on our website where you can find more information about this project. We have quite extensive information on what this project is all about. Next slide, please. So for the goals for today, next slide. I'm just providing a little bit of background here. So we all agree that we need additional PrEP products to meet the needs of everyone. All people requiring PrEP should have the adequate and appropriate product for them. But the development of new PrEP products is quite challenging. One part of this is the trial design. How do we demonstrate efficacy and safety? The other part that contributes to the challenge is the ever-changing context of the HIV epidemic, increasing use of already approved PrEP products, which are quite effective, and of course, increase in HIV testing and referral to treatment. And of course, in terms of drug development, the whole schema of regional and national regulatory requirements can also provide quite a few challenges in terms of bringing these drugs to market. So we do need global cooperation and collaboration to ensure success, and we want to support that with today's session. Next slide, please. So in today's session, we basically have three main points. We will start with reviewing the need for innovation and discuss what some of the potential approaches could be. And we will talk about the counterfactual placebo estimate for primary efficacy analysis. David Glidden will lead us through that. And then we will talk about how a recency assay that we are often talking about in terms of building the counterfactual is actually being used around the world today, and how this could contribute to the estimate that we're looking for. And that will be presented by Jessica Justman. Then we will follow up with two presentations, so two different test cases of phase 3 programs, the islatravir and lenacapavir programs, and outlining for us to see how this approach is actually working in real life. These programs are being implemented as we speak. And then we will finish with a multidisciplinary discussion of the challenges and opportunities in trial implementation. Next slide, please. I want to alert everyone that we actually have an abstract presentation as well, which will go into much more technical details about the recency assay. You can see that presentation, the poster, as well as the pre-recorded presentation. Next slide, please. I'd like to now acknowledge some of our key partners. Next slide. So very special thanks to the Project Steering Committee, also the Public Meeting Planning Committee, our Recency Assay Working Group, and the Prevention Trials of Women Working Group. And again, I put in the link where you can get much more information about these parts of the project. Next slide, please. These are the members of our steering committee. I'm not going to go through it one by one, but you can see it is extensive and very representative of the different stakeholders, as is characteristic for the forum. Next slide, please. We want to have a special thank you to the Bill and Melinda Gates Foundation for funding this project, as well as our pharmaceutical and diagnostic companies who are also supporting the overall project. Next slide, please. Then last but not least, of course, the forum team. Tamar Chalitze is our HIV team lead. She and her team, Joseph Lau Tinkani and Nick Murdoch, have done an amazing job in preparing for this. And a special shout out to Nick, who took on the major responsibility to work closely with Tamar and myself to make this happen. And then, of course, we also have other people in the forum. Brenda Rodriguez is our deputy director. She also helped a lot here. And Victoria Mason, she is a research associate. Next slide, please. So now it is my great pleasure to introduce Dr. Deborah Burnkrant, who's going to make some opening remarks. And she is the director of the Division of Antivirals at the US FDA. She served in that role for 20 years. In this capacity, she oversees the regulatory review and related processes for HIV and, of course, all other viral diseases. And we're so fortunate and happy to have you with us today, Deb, so that you can make some introductory remarks regarding your view of where we are with this whole challenge that we're dealing with. Please go ahead.
Thank you, Veronica. Good afternoon, everyone. As you know, we are currently living in an era in which two highly effective FDA-approved drugs are available for the prevention of HIV infection. And while this achievement is great news and has been a boon to efforts in combating the HIV epidemic globally, it also poses challenges to the study of new PrEP agents, which we agree are greatly needed. All of us at FDA acknowledge that the traditional approach of conducting randomized controlled trials for HIV prevention may no longer be suitable. For one, conducting placebo-controlled trials in the setting of approved and available PrEP products is not ethical. The alternative, to include approved PrEP drugs as controls in superiority or non-inferiority design trials, also has inherent challenges, such as the risk of not being able to demonstrate statistical superiority or non-inferiority due to the low number of observed HIV infections and the need for very large sample sizes that are often infeasible. With the help of the Forum for Collaborative Research over many years, including numerous working groups and public meetings, a new proposal was developed to address the challenge of studying next-generation PrEP agents. Out of these meetings and working groups, the use of a counterfactual estimate of HIV incidence has been put forth as a comparator to evaluate the efficacy of new PrEP products. We will be hearing about this approach in more detail today with a presentation from Dr. David Glidden. FDA supports the use of a counterfactual estimate of HIV incidence, but recognizes that such an approach for externally controlled trials has not been fully tested to date to support a drug approval. We also recognize that this approach may have some limitations and uncertainties. Given these issues, we have worked with drug developers to incorporate more than one method into their trials to estimate the counterfactual HIV incidence, including the use of HIV-1 recency assay testing, which will be discussed today by Dr. Jessica Justman. Other methods of estimating the background HIV incidence may include epidemiologic estimates from trial sites or similar geographic regions, the incidence of acute HIV infections during trial screening and enrollment, and potentially the adherence efficacy relationship of the active comparator. If several of these methods show similar estimates, then we can be more confident about the comparisons made and the ability to use these data to provide persuasive evidence of a treatment effect and interpret efficacy findings for new agents. To provide additional reassurance regarding the validity of the counterfactual HIV-1 incidence estimates, however, and to assure internal consistency within the randomized framework, the inclusion of an internal active control in PrEP trials is also recommended. The two investigational products you will hear about during this satellite session is Latravir and lenacapavir by our industry colleagues incorporate these new methodologies into their phase 3 trials. We support the design of these trials, and while recognizing the potential challenges with interpreting the overall efficacy results, especially when HIV incidence is low, we believe it is time to implement new methodologies so that new and novel PrEP products can be developed in an accelerated time frame, and people at risk can have highly effective choices for HIV prevention that fit their individual preferences. An important and welcome step forward would be the real-time collaboration among all stakeholders to better understand the validity of the counterfactual estimates using recency assays and other methods, and to review the lessons learned after the trials complete to better inform future studies. Lastly, all stakeholders, including the FDA, the HIV community, and researchers, need to continue to explore scientifically and statistically sound pathways to convincingly determine the efficacy and safety of new PrEP products. Thank you, and we are looking forward to today's presentations and panel discussions. Excuse me. Back to you, Veronica.
Thank you so much, Debbie. That was very, very helpful overview. And it is now my distinct pleasure to introduce another colleague from the US FDA, Stephanie Troy. Stephanie Troy is an infectious disease specialist who has been working as a clinical reviewer at the Division of Antivirals since 2016. She was an assistant professor at the Eastern Virginia Medical School before that, where she saw patients and also conducted clinical research on polio vaccines and vaccine responses in people with HIV. So Stephanie, thank you so much for co-moderating with me. Welcome on board. And I will now hand over to you to introduce our first speaker.
Thank you, Veronica. Can you hear me?
Yes.
Okay, wonderful. Thank you. It is my distinct pleasure to introduce Dr. David Glidden, who will be giving an overview of the rationale and approach for counterfactual-based placebo estimate. Dr. David Glidden is a professor of epidemiology and biostatistics at the University of California at San Francisco. His work focuses on prevention and treatment of HIV infection. He has been involved in studies of HIV prevention for over 12 years, and his current work involves studies of the global scale-up of PrEP, innovative designs and statistical analyses of new PrEP agents, and pharmacologic markers of PrEP exposure. And for my own part, I read a number of his papers, which have helped me with my review work at FDA. So without further ado, I introduce Dr. David Glidden.
Good day. I'm excited to follow on Dr. Bernkrant. I'm going to elaborate on a number of the issues that she raised in her talk. In particular, I'm going to talk about a counterfactual-based approach to learning in PrEP trials, the concept, their use, and a little bit about strategies for calculating a counterfactual. Next slide, please. As Dr. Bernkrant had said, over the last 5 years, the pivotal trials of PrEP agents have been ones which have been active controlled. They've employed a double-blind, double-dummy design where people have received 2 possible agents, one of which is an active for the control and one of which is active for the investigational. And then they also receive placebo for the non-corresponding randomization assignment that they have. Now, this has been a strategy that's been adopted by the HPTN 083, 084 injectable cabotegravir studies, as well as the Discover study of FTAF for PrEP. The complication with this design is it works well if we have clear superiority or clear non-inferiority, neither of which we can count on going forward. This design has a very important positive feature that I want to point out, and that is we have randomized comparisons of 2 groups which are treated the same way during the trial because they're blinded to their assignment. Next slide, please. So as I discussed the idea of a counterfactual, we're imagining a scenario that doesn't exist. And I think of it in a very specific way. And that is, I imagine that such a study would have a 3rd assignment where people would be randomized along with the investigational agent, the control, to a scenario where they receive 2 products, and both those products would be the placebo for the control product. and for the investigational product. And such a scenario would have the advantage that, again, by randomization, we would be comparing similar populations. By the blinding of the products, there would be similar treatment or Hawthorne effects during the trial, and the control group would be a concurrent one. So it's obviously a thought experiment and not an ethical thing to do. Next slide, please. I want to point out how valuable a counterfactual arm could be in the interpretation when we have the scenarios that we expect in the future. That is, it's possible that we may have two possibly effective agents and observe low number of seroconversions on two arms and possibly low rates of HIV acquisition during the trial, also on two arms. And so here I've got data based on the Discover trial of a scenario where there's a small number of incident HIV infections during the study. And under a scenario where we believe that the counterfactual HIV incidence rate, which I have here as 0.3 per 100 person years, in that case, the Discover trial would look quite grim with 54% efficacy on the investigational arm and 15% efficacy on the control arm. In contrast, that same data on the next slide, please, if there were a moderate to high risk of HIV acquisition on a counterfactual placebo arm, something like 3.1 per 100 person years, we'd see that there'd be a low number of HIV infections observed on each arm, but we would expect a large number on a counterfactual placebo arm, telling us a large number of HIV infections have been prevented on each arm, and that both arms are showing substantial efficacy. So what I want to stress here is how important, in the situation that we don't have clear superiority or clear non-inferiority, that a counterfactual placebo arm could really disambiguate the trial for us. Next slide, please. So a number of strategies, and Dr. Bernkrant mentioned this, have been developed for how we might infer the counterfactual, or I here call it the B or background HIV rate. All of these involve leveraging data collected as a part of the trial. That's the auxiliary data column I hear, and that might include demographics, reports of sexual practices, diagnosed STIs, information about where and when the trial is being conducted, or possibly drug levels or other adherence information. This is then combined with historical information, perhaps from previously completed trials or cohorts, or things that we know about the properties of assays for drug levels or for recent HIV infection. And that this is then used to leverage and give us a strategy for calculating the possible counterfactual placebo. And as you can see, it's been a fairly active area of investigation. Next slide, please. I want to focus on the next slide, please, on a particular example and introduce the concept of recency assays that Dr. Justman will discuss. The idea with recency assays is to use something that's been done in HIV surveillance in populations for a long time, which is to use cross-sectional testing to infer HIV incidence. And the idea is that in the populations that might enter a PrEP trial, we might incorporate recency testing as a part of the HIV testing, which is conducted during the screening period. And one of the things that Dr. Fei Gao has shown in her careful interrogation of recency assays is this allows us to reconstruct something that's like a pre-trial cohort coming into the PrEP trial. And this allows us to estimate HIV incidence in the period covered by recency just prior to entry into the PrEP trial. So this gives us a similar population to that which is enrolled in the trial, while not quite identical, and while not exactly concurrent, it does give us an adjacent time period. On the next slide, please. It does come with some important complications, which statisticians have begun to admire, if not entirely solve. The first is that there are several possible assays and algorithms for determining recency. And from a statistical point of view, we need to understand the properties of these tests because they are not perfect and they involve some degree of misclassification in a loss of recency among some recent infections, as well as false recency among some infections of long durations. And a critical issue in calculating this incidence estimate is the correction for these test properties. And then the second is the challenge that comes with incorporating these into our trial designs. And that is, to date, when we've screened our participants for trial designs, our emphasis has been on validity, yes, but also on the best yield of possible participants who will be eligible for our trial. And there's been some degree of pre-screening involved, perhaps even pre-screening on HIV status. Once we use recency as a part of the screening process, some tensions arise between that pre-screening and also recruiting what would be a representative sample of people in the population at risk for HIV infection that would be otherwise eligible for the trial. In addition, other practical issues include incorporating the history of HIV testing for participants, which may be included in the recency period, and also prior PrEP use as well. So I find myself greatly in agreement with what Dr. Bernkrantz said, and I wanted to share with you a little bit of the developments and thinking about counterfactual placebo incidence calculation, as well as particularly the value of recency. Thank you.
Thank you, David. I can't think of anyone else who can walk us through very complex, dense material in such a way that even I can understand it. So thank you very much for that. Next, we will have Jessica Justman, and she's the Senior Technical Director at ICAP, which leads the population-based HIV impact assessment project in PEPFAR-supported countries. She's also the principal investigator for the Tracking with Recency assay to control the epidemic, the TRACE initiative. So Jessica, wonderful to have you here. Thank you so much.
Well, thank you very much, and good afternoon and good evening, everyone. I'm really pleased to have the opportunity to talk to you about HIV recency testing in routine HIV testing programs. And really, the overall purpose here is to just give you all an idea about how much recency testing is already happening. Next slide, please. A few definitions first, just to make sure we're all clear that the recency testing that I'm going to talk about really builds on, as Veronica mentioned, the limiting antigen or lag-based testing technology that has now been developed into a rapid point-of-care HIV test that can distinguish between a recent, and we define recent as in the prior 12 months, from a long-term infection. When used with blood, the recency tests strips, as shown in the photo, will reveal up to 3 lines. And if you look at the test strip all the way on your right, you can see there's a control line at the top, a verification line in the middle, and a long-term infection line at the bottom. that indicates that this blood sample came from an individual with a long-term infection. If you move over to your left, you see a couple of test strips that have only two lines, the control line and the verification line, and those are consistent with blood samples that came from individuals with a recent infection. So it's the absence of that long-term line that indicates a recent infection. You'll hear me refer to RTRI, which means rapid test for recent infection. And then also RITA, or ITA, stands for Recent Infection Testing Algorithm. There are many different algorithms, but in this particular context here, I'm going to talk about the algorithm that incorporates viral load with an HIV RNA over 1,000 when combined with a positive RTRI infection. That would indicate somebody who had a RITA recent infection. Next slide. Tracking with recency assays to control the epidemic, or TRACE, is the name of the project that began just at the end of 2018 to establish in countries nearing epidemic control a recent infection surveillance system to detect, characterize, monitor, and allow for interventions in real time for recent infections that occur among people living with HIV who have been newly diagnosed in routine clinical care settings. TRACE is funded by PEPFAR through CDC, ICAP as well as UCSF are the global partners, and there is a website with a great deal of information on it, and I've provided the link there for you on the right side of the slide. So I certainly encourage you to take a look there at the TRACE website. Next slide. Where is recency testing happening? Well, it's really happening across the globe. This map shows the location of the over 30 PEPFAR-supported countries that have or will establish recent infection surveillance systems. In the past quarter, these 30 countries have conducted over 30,000 recency tests. So that's a quarterly number. 16 of these countries are supported through TRACE. 8 are supported by UCSF, 8 are supported by ICAP. ICAP support is at 1,600 HIV testing sites, and I just emphasize these numbers really to give you a sense of how much capacity is being built on recency testing and how much has been accomplished in the last 2.5 years. Next slide. Recency testing really looks quite different from country to country. And here I am showing you the different models of recency implementation and how they really depend on these 4 design elements, these 4 core components. The first is recency classification. We've talked a little bit about that already in terms of whether viral load is part of the algorithm. And this is the viral load taken at the time of HIV diagnosis, and it helps us reclassify people who've come in to retest who are often on ART and who are virally suppressed. The second core design element is the location of the testing. Does it happen at the HIV testing service site? And is it conducted by these HIV testing service counselors right at the time of diagnosis? Or is the blood sample taken from the site and sent over to the lab for recency testing? The third component is consent, and this is a reminder to tell you that under the TRACE program, all recency testing is conducted as part of a research protocol because the recency testing at this point is not a type of test that's approved, and it's certainly not approved for individual use. It's intended to be used as part of surveillance. And the approach to consent, whether it's written or verbal or opt-out, does really impact the uptake of recent infection testing. And then finally, the fourth element is whether the results are returned to clients or not. And the majority of countries are not returning the results to clients and are using it for surveillance only. There are some countries that are returning results, but they also use the results for surveillance. Next slide. Here we see the recent infection surveillance cascade. And this is just to remind us that the data that we see, and that I'll show you in a moment on recency testing, it really only represents a subset of individuals living with HIV and only a subset of those who were newly diagnosed, because these have to be individuals who are seeking care, receiving testing, they're eligible for RTRI testing, and they agree to have it, and the results are collected. And this is not done at all sites. It's done at a subset of the sites that offer recency testing and report their data. Next slide. So here are some data just to make things easier. We're not showing the country names, but these are all sub-Saharan African countries. Listed across the bottom is countries A through H. And you can see there's quite a variation in RTRI uptake. And to make that easier for you to see, let's just take a look at country A over on your left, where there were 892 individuals who were newly diagnosed with HIV. In that country, excellent uptake. 100% of those newly diagnosed individuals underwent recency testing. And of those, 8% were RTRI positive. So we really look to see an uptake level of about 80%. A number of these countries are close to or slightly above 80%, but others, such as country E in the middle, only have 12% uptake of RTRI testing. And then the range of RTRI results ranges between 8 and 19%. 8 we saw in country A, and then in country D, it's up to 19%, but that range is what we would expect for RTRI results. Next. Now, Dr. Glidden talked a bit about the importance of using algorithms to reclassify results because of situations that impair really the specificity of the testing. And we talked before just a few moments ago about viral load. So here are 4 countries, data from 4 countries that are using the RITA algorithm. And in each of these countries, a very substantial proportion of RTRI recents are reclassified to long-term. Again, looking at country A as an example, 56 RTRI recents were reclassified to only 31 RITA recents. So just keep that proportion in mind. Approximately 1/3 of RTRIs need to be reclassified as long-term, and that leaves the remainder as recent infections. Next. So here we can look at some data on countries that are using the RITA algorithm. And again, if we just look at country A over on your right, 97% of those who were newly diagnosed were tested with a full RITA algorithm, and 4% of those individuals were RITA positive. So here the uptake varies all the way from 11% to 97% across these 4 countries, and the RITA recent results varies from 3 to 5%, which is in the range that we do often see. Next slide. Here we can drill down on one particular sub-Saharan African country to just get a better sense of who demographically are accounting for recent infections. And what we can see in the orange bars are adolescent girls and young women between the ages of 15 to 19 and also 20 to 24 together. Each have high proportions of RITA recent infections, about 10 to 12% each in those age groups. Males in the green bars between the ages of 20 to 24, about 8% of recent infections are found in that demographic group of young males. Next slide. We might also want to know what's the distribution of recent infection by point of testing location. And here the green bars show the total number of RITA recent infections, so different than in the prior slide. And the black diamonds show you the percent of RITA recents out of the entire total. So the black diamonds for OPD of about 25%, and for index testing, also towards the middle, another 25% show us that about half of recent infections are identified in the outpatient department and index testing programs in this country. Next slide. And then almost finally, the data are also used for real-time surveillance of recent infection trends and clusters through dashboards, such as the one there on your left, where you can see the number of recent infections in the red dots as compared to the number of long-term infections in gray dots. This slide was shown actually 2 years ago by our colleague from the Rwanda Biomedical Center at IAS in Mexico City. And over the right-hand side, you can see a chart that just shows RTRI results from 5 facilities that had the highest proportion of recent HIV infection. And those 5 facilities then were targeted for specific interventions. Next slide. And then, of course, the very important use of recency data is to estimate HIV incidence among HIV testing service clients. Now, I'm not going to talk about that. We heard about it in some detail from Dr. Glidden. Just to say that back calculation and CD4 diagnosis delay models have been used by CDC to use recency data and come up with incidence estimates. On the trace side, we have been exploring the use of stratified extrapolation to use the recency data and weight each recent case to compensate for recently infected people who weren't tested. This particular activity got a little bit pushed to the side during COVID, but we are about to return to it now. Next. So just to summarize here, I think I've given you a picture that shows how over 30,000 recency tests are being conducted per quarter with point-of-care rapid testing technology in PEPFAR-supported African and Central American countries. Capacity is rapidly expanding. It's very important to use the RITA algorithm to improve the classification and precision of estimates. And the methods of using program data on recency to estimate HIV incidence is certainly of great interest, and these methods are in development. Next slide. Just to say thank you very much and to acknowledge Dr. Suzue Sato and my other TRACE colleagues at ICANN.
Thank you very much, Jessica, for that wonderful overview of how this assay is used and the kind of population level data and what surveillance data and public health tool it can be. Now, it is my pleasure now to introduce the moderators for the next panel. And first, we have 2 moderators. I will introduce Helen Rees. Helen Rees is the Executive Director of the WITS Reproductive Health and HIV Institute at the University of Witwatersrand. Dr. Rees is also the chairperson of the South African Health Products Regulatory Authority. She's recognized for her expertise in HIV prevention, reproductive health, and vaccines, and has served on numerous national and international structures. We also have Ken Mayer, who is the Medical Research Director and Co-Chair of the Fenway Institute. Dr. Mayer is also a Professor of Medicine at the Harvard Medical School. He's highly recognized for his contributions to international HIV/AIDS research, LGBTQ health, and HIV/AIDS treatment and prevention. So Ken, I'm handing this over to you.
Thank you, Veronica. Good afternoon, good evening, everyone. It's a pleasure to introduce this next session. We're going to have 2 parts to this. We're going to have 2 speakers, Drs. Mopli Das and Mike Robertson, who are going to provide insights into their phase 3 programs, very innovative use of the counterfactual placebo estimate as a primary efficacy analysis. And then we will have a panel to really further develop the themes that Mopli and Mike will elaborate on. Dr. Das is Executive Director of Clinical Development in the Virology Therapeutic Area at Gilead Sciences. She is trained in infectious diseases with a background in epidemiology. Dr. Robertson has extensive experience in HIV prevention. He has worked in HIV vaccine trials. He is currently Executive Director of Infectious Diseases at Merck, and his research interests include vaccines and infectious disease development. And he's currently leading this Latravir HIV PrEP clinical trials. So Dr. Das, would you like to start and talk to us about lenacapavir studies that you've been planning? Thank you.
Thank you, Dr. Mayer, for that lovely introduction. And I would like to thank the forum leadership, particularly Veronica and her entire team, for bringing together sponsors, regulators, clinical scientists, and statisticians to help us develop this trial to allow us to evaluate new products for HIV prevention. It's my honor to present on behalf of our investigators, our global community accountability group, including our member Natando Yola, who's here with us today on this call, our team internally about how we designed, operationalized, and implemented these studies. So I will get going. Okay. So Dr. Bernkrant and Dr. Glidden already went through the overview that leads us into this topic of the challenges in existing PrEP clinical trial designs. So we're using the novel counterfactual background HIV incidence design in our lenacapavir and FTAF evaluation program for prevention. Our novel HIV counterfactual incidence design is based on the Pearl Index that Dr. Jeff Murray presented at the International AIDS Conference in Mexico City in 2019. And here you can see, if you know the relative incidence of pregnancy in a group of fertile women, let's say it's 80 per 100 person years, and you have a single arm study where you put fertile women on a new contraceptive, and you observe only 1 or 2 pregnancies or contraceptive failures, you'll know that your new contraceptive is working well. We can translate this into HIV, where if you know what the background incidence is in the communities where you want to study your novel HIV incidence, a novel study drug, you can put people on that study drug and see if you're able to lower the HIV incidence on the study drug. So that's one of the critical aspects of this design, is to have a robust enough background incidence in the locations that you're doing the study, and that's part of the considerations as we plan these trials. As Dr. Glidden alluded to, as well as Dr. Bernkrant, counterfactual HIV incidence is also useful in the interpretation of classically designed trials. In Discover, we evaluated the background incidence using a variety of methods. Because the incidence was so low on study drug, people may have been concerned if that incidence was low due to the low risk within the population or low risk in the community where we studied the drug versus the efficacy of the drug. And from all of these estimates, using the different methods summarized by Dr. Glidden, Dr. Bernkrant, we saw that the incidence was quite robust in the background in the sites where we did the study, suggesting the efficacy of both of the products, FTAF and FTDF, in our trial. There's a number of considerations that we need to take into account as we look at doing these new types of trials. One that was alluded to is the choice of the recency assay. There are many different tests that evaluate recent infection, and they have different test characteristics. The other piece is the choice of the algorithm, or RITA, that Dr. Justman referred to. There are different ways to do the algorithm, and all of those things need to be considered. It's really critical that we engage with community, people living with HIV and who may benefit from PrEP, civil society, investigators, and all these other key stakeholders before the trial design stage. We also have to consider, when we're looking at the counterfactual estimation, is this pre-randomization or post-randomization, because secular trends can affect HIV incidence over time. We have to choose sites that have a significant background HIV incidence to support the design, as I mentioned, and we have to introduce an incidence cohort prior to the randomization cohort and make sure that the incidence cohort characteristics are similar to the randomized participants. We have to set the statistical criteria for evaluation of clinically significant efficacy. And we have to consider triangulating alternative methodologies for estimating counterfactual HIV incidence to ensure that recency is performing as expected. So in our new prevention drug development program, we have 2 trials. Purpose 1, which takes place in cisgender adolescent girls and young women, and Purpose 2, which takes place in cisgender gay men and other men who have sex with men, transgender women, transgender men, and gender non-binary individuals. We are evaluating lenacapavir and FTAF in Purpose 1 and lenacapavir in Purpose 2. So Purpose 1, sorry, I advanced twice. Purpose 1 is our trial, and you can see what the acronym stands for here. Next slide, here we go. Okay, so as I mentioned, we have the cross-sectional HIV incidence cohort where everyone will be screened for HIV, and if they are positive, they will have a recency test done to see if they have evidence of recent infection, and then we will back calculate HIV incidence based on the readout. And then they will be, those people who are negative will be randomized 2 to 1 in either a lenacapavir arm, an FTAF arm, or a Truvada arm to observe the incidence on those products for 52 weeks. This trial has 2 primary endpoints. We're evaluating both lenacapavir and FTAF for prevention in cisgender women. So the cross-sectional HIV incidence using recency will be the estimate that is the primary comparator for our endpoint. However, we will also explore alternative ways to calculate that counterfactual, including the methods that Drs. Glynn and Bernkrant summarized earlier. In Purpose 2, you can see what the acronym stands for here in bold. This study is in a 2-arm study. Again, we have that cross-sectional HIV incidence cohort to estimate the background HIV incidence. And we will have a lenacapavir arm, again, randomized 2 to 1, excuse me, with Truvada or FTDF as our internal control. And we will use multiple methods to evaluate the counterfactual in addition to the recency assay. This has one primary endpoint, which is the incidence in lenacapavir compared to the background HIV incidence as estimated by the recency assay. So I would like to now turn over back to Ken to turn over to Dr. Robertson. Thank you very much for the opportunity to present.
Thank you, Dr. Doss. And now it's time for Dr. Robertson. Thank you.
Thank you, Ken, and thank you, Dr. Doss, for that nice setup. And I also want to thank the forum for organizing this meeting and also the series of workshops that have led up to this approach. What I'd like to share with you is the overall clinical development plan for monthly oral islatravir as PrEP. This just shows the studies that are part of our clinical development program. We have an ongoing phase 2 trial, which is protocol 16, which I won't go into detail discussing today. Actually, Dr. Sharon Hillier will be presenting an updated summary of data from this trial on Tuesday at the late breaker oral abstract session. But I want to emphasize that this phase 2 trial is really critical for the development program because in this trial, we have, these are low-risk individuals, but we have a very extensive PK analysis in both men and women from this trial, including extensive tissue PK. And what we are hoping to demonstrate in this phase 2 trial is that we get good exposures in both men and women at all of the relevant sites for HIV transmission. So please refer to that oral abstract session later this week to get detail, and we'll be presenting the tissue data at a subsequent meeting, hopefully in the near future. Today, I'm going to talk more about our 2 ongoing phase 3 trials. These are the Empower program. We have one phase 3 trial called Empower 22, which is in cisgender women. I'll talk more about that in a subsequent slide. And then we have another trial called Empower 24, which is in men who have sex with men and transgender women who have sex with men that are at high risk of HIV infection. And one of the things I want to emphasize is that we are using this counterfactual estimate approach in both of these trials, but they are different, and I'll emphasize that on the subsequent slides. the Empower 22 trial. As I mentioned, this is in cisgender women. And this trial is going to be approximately 4,500 participants, 90% of which will be enrolled in Southern and Eastern Africa. The other 10% will be women in the United States. In this trial, and it's a double-blind, double-dummy trial, there is an active control. Women are randomized to get either monthly islatravir and a placebo to Truvada, or they get active Truvada and once a month they'll take a placebo to islatravir. Now, this trial, the primary efficacy endpoint is actually the comparison between the incidence rate in the islatravir arm and the Truvada arm. So this is really more of a traditional superiority trial. As Dr. Glidden and Dr. Bernkrant talked about earlier, this is very similar to some of the previous trials that have been done, such as the HB10-084 trial, where the real comparison is to show superiority of islatravir to the active comparator. So this is a much more traditional trial, but we do have as a secondary endpoint this comparison to the background incidence rate, and we're using this recency-based approach that has been described to estimate that background rate. But again, I want to emphasize that the primary endpoint of this trial is the kind of more traditional superiority trial. So this next slide just shows schematically how we do this, and you'll notice it's very similar to what Dr. Das was showing for the lenacapavir trials, where we have a period where we're screening individuals. Those that are HIV seropositive at screening are go on to be assessed for to see if they are recently infected, and those that are, that's used to estimate the counterfactual background incidence rate. Those that are negative get randomized to either of the active groups, and then there is kind of a very standard follow-up where we do this. So again, we are using this counterfactual approach as a secondary endpoint in this trial, but the primary endpoint is the more traditional approach of just showing superiority of the investigational drug, islatravir, to the approved product, Truvada. Now, by contrast, the Empower 24 trial, which is our trial in men and transgender women who have sex with men, is very similar to the design that Dr. Das was talking about for the lenacapavir trials. You'll notice that it's a smaller trial, only 1,500 individuals total. They're randomized in a 2-to-1 ratio to get either monthly islatravir plus a matching placebo to either Truvada or Descovy, or they would get the active Truvada or Descovy and a matching placebo once a month to islatravir. Here, in contrast to our trial in women, the primary endpoint really is this counterfactual based approach, where we're looking at the estimated background rate as estimated by the recency assay, and our hypothesis would be that the incidence rate in islatravir arm is lower than that estimated background rate. We will have, as a secondary endpoint in this trial, the direct comparison between the arms, but the primary endpoint will be the counterfactual approach. And this final slide that I have just shows, again, schematically the design of this trial. Again, people are screened, and those that are positive are assessed to see if they were recently infected, and that group is used to estimate the counterfactual background incidence rate. Those that are negative can be randomized to one of the active arms. So this is our program. And again, what I'd like to just say is that, we really look at these as a package. So to begin with, we have our phase 2 trial, which is evaluating safety and importantly PK, including tissue PK in both men and women. And we really view that as kind of the link between these to show that we are getting the product into the right places at the right time. And then we have a more traditional trial in women, which is a traditional superiority trial. We will be assessing the counterfactual in that, but it's really a secondary endpoint. And in the trial in men and transgender women, that counterfactual approach is the primary endpoint. And what we hope to show is that the combination of all these trials, including the robust PK data that we have in our phase 2 trial, really does say that we can help bridge the efficacy that we would observe in that more traditional trial in women to the broader population. And we think that these data will, really give that full package for demonstrating the efficacy. So we are using this counterfactual approach, but one of our pivotal trials really uses a more traditional approach. And then we have this robust PK bridging that we're hoping to link the two together. So with that, I will stop and turn this back to Ken and Helen.
Thank you very much. Because of time, I'm going to take the opportunity to thank both Mike Robertson and Mupali Das very much for their case studies, which we're now going to discuss in a panel. Unfortunately, time is tight, so we won't be able to take Q&A from the audience, but I will pose a question each to each of our panelists. I'll introduce the three speakers. Stephanie Troy is an infectious disease specialist who's been working as a clinical reviewer in the Division of Antivirals at FDA since 2016. Prior to joining the FDA, she was assistant professor at Eastern Virginia Medical School, where she saw patients and conducted clinical research focusing on polio vaccines and the vaccine response in people living with HIV. Our second panelist is Totlang Setloho, who is Senior Manager of Clinical Evaluation Management at the South African Health Products Regulatory Authority. He has a background in both pharmacy and epidemiology, and his experience includes clinical evaluations and policies and guidelines, as well as clinical trial management and vigilance policies and guidelines. The third speaker is Ntando Yola. Ntando is a community engagement lead at the Desmond Tutu Health Foundation. He's been with the development and implementation of community engagement strategies and forming partnerships with key stakeholders to advance HIV prevention research. So I'm going to launch straight in. Stephanie, I'm going to pose the first question to you, and clearly we have two regulators on this panel. Stephanie, first of all to you. As a regulator, what will you be watching for as these test case phase 3 programs based on counterfactual placebo estimates evolve and report out? And I'm going to ask you to be very succinct. Stephanie.
Thank you. So the main thing that we will be watching for is consistency in results between estimates based on the recency assay and estimates based on at least one other method for estimating the counterfactual HIV incidence rate. And this will be important not just to provide confidence in the estimate, but also to provide assurance that the background HIV incidence rate is substantially high. And we're looking for a rate at or above 3 infections per 100 person years. And that will be important for the secondary analysis, since as was previously mentioned in the two talks, these studies will also have an active control using an approved PrEP product as a secondary comparison to support the primary analysis. Now, as has been mentioned, this can't be done as a non-inferiority trial due to feasibility issues. However, if the background HIV incidence is sufficiently high, then an HIV incidence rate for the investigational drug that is similar to or lower than the rate for the active control would provide reassurance of efficacy. And the success criteria also have a built-in buffer to account for potential uncertainties in the external controls. I want to emphasize here that while using the counterfactual placebo estimate for HIV-1 PrEP trials is a creative novel approach, this approach was built on statistical analyses of available data. And we will be relying on the sponsors and investigators, including Dr. Das and Dr. Robertson, to proactively collaborate and share their experiences so that we can all learn from each other and refine this approach moving forward. Thank you.
Thank you very much. I'll go straight to the next question for Totlang Setloho. What are the needs for a regulatory authority like SAHPRA to address concerns that you might have using the counterfactual placebo estimate approach to assess efficacy of new PrEP products?
Thank you, Prof. This is a new approach that we'll have to wrap our heads around, and it's something that departs quite drastically from our classical RCT study design, and some of the issues that we may have. Obviously, we may not have problems with sample size calculation methods and the related assumptions, and both samples are obtained from the same community anyway. So we don't expect a lot of issues there. But the main issue that we may raise would be a perception of selection bias. And it's something that we will have to discuss with the sponsor in terms of us better understanding whether there's a need for a measurable justification where the counterfactual placebo group, which is already infected, is considered to have similar baseline characteristics to the experimental group. Baseline characteristics such as social behavioral traits, comparative propensity for risky practices, and other potential determinants. So we may need some clarification there, whether we are on the same page or we are understanding the issue differently. And the confounding, potential confounding is one of the issues that I would need clarification on. And it is linked to selection bias. Lack of classical randomization amplifies the impact of unobserved time-varying confounders on the derived effect size for clinical efficacy of these PrEP intervention, and that occurs in very unpredictable ways, of course. Alternatively, the assumption of linearity and parallel trends between the counterfactual placebo and the treatment periods need to be validated and justified. We are aware of various complex methods, mathematical and statistical methods, to correct the violations of such assumptions in observational studies, but we need to confirm validity for clinical efficacy assessments. We need a motivation in the proposed protocol to get us over that hurdle. Overall implications for the strength of validity of the results, gray area, and in which direction will validity or strength be affected. We don't know. We'll need clarification from the sponsor. Finally, we'll need to understand how the chosen exclusion and inclusion criteria solve for confounding and increase precision of the estimate in the absence of concurrency and randomization, for instance. So yes, we support improvements in research methods, especially where RCTs may not be feasible for the reasons already stated. But it's a journey that we'll have to take together with the sponsors and get to a stage where we are all on the same page as far as the counterfactual placebo estimate method is concerned. I think those would be the key issues that we will raise during our meetings and interactions with the sponsors as far as this new design is concerned for clinical efficacy assessment. Thank you very much, Chair.
Thank you, Tatlang. And I would just add here that I think dialogue between regulatory authorities would also be extremely helpful and would encourage FDA, SAPRA to find an opportunity to do that. Thank you very much. Our third speaker is Ntando Yola. So Ntando, you have a lot of experience in advocacy and community voice representation. What has helped you navigate the issues on PrEP clinical trial design and discussions on basing the primary efficacy analysis on the counterfactual placebo estimate? From your perspective, what are the responsibilities of the study sponsors and the regulators with respect to responsible use of the data? Ntando.
Thank you very much, Helen. And I think top of everything, I think the field has really come a long way to where we are. I mean, it's evident with the results that we are seeing and all the products that we have actually for HIV prevention, which are actually putting us in the space that we are in with regards to, that's really making us having this discussion about a need for new trial designs. And what has really helped, in fact, what is helping is what has really helped previously about how organized a community of advocates and communities are, from a global level to regional, national, and even at a local level. And these are important groups that really need to be recognized and approached to be able to, in fact, brought into the processes as early as possible, and as it has been. In fact, from what we've heard, for instance, about the work that Gilead and Merck are going to do, this is exactly one of the things that they've done. Is I think as we have heard from them. Now, one of the things that is important, to highlight as part of this, that given that the field has really come to accept, embrace the process of bringing communities early on into the processes of research and development, especially around HIV, ensures that there is a sustainable approach into making sure that communities and advocates are part of these processes. Even when new developments come into the fore, they basically are not, there are no gaps between engagement with what has happened previously and what is happening today and even the future. That sustainability is important. And that is actually a role that speaks to part of the question, where we really make sure that research, the engagement of communities and their diversity is not specifically pinned to specific trials. We have an overall value chain, a support of all these efforts that are happening at all of these, the different levels that we, where research is happening. And I think that's one of the important things, which I think is very key for funders specifically. But I think one of the things that you've also spoken to, Helen, is the importance of regulators speaking to each other, be it at international and at a national level. But in fact, I would really like to add there that one of the things that we see is that regulators engage with funders, and then each of these engage with communities and advocates separately. And I think it is also important, because this seems to be a gap where we have these conversations as collectives. In fact, these conversations, if they happen in that nature, they're even able to address the issues that we are talking about with regards to, having communities that are very informed and capacitated, in fact, are able to play a very important role of accountability, especially particularly for our discussion with regards to use of data in a very responsible way. So I think I'll leave it at that for now.
Well, thank you very much. And I apologize to all the panelists that were pushing you so much, and our other two speakers for not being able to come back to you because of time. But on behalf of Ken and myself, I'm going to now hand over to Veronica, who will take us into the next session. But clearly, there's going to be a lot more discussion about counterfactuals and this novel way of in which we are going to have to deliver clinical trials in the future. More understanding, as you say, from a community level as well. But thank you very much to our panelists. And Ken, thank you very much for being the co-moderator. And I will hand straight back to Veronica Miller. Thank you.
Thank you so much, Helen, and thank you for the panelists for this last session. We now want to just engage in a little bit of a broader discussion, and I'm so pleased to have as my co-moderator for this last session, Rachel Bagale from the WHO. She is the unit head for testing, prevention, and populations, global HIV, hepatitis, and STI programs at the WHO in Geneva. She led, together with colleagues from UNAIDS, the most recent updates with regard to the Ethics and Prevention Trial Guidance Report from the WHO and the UNAIDS. So Rachel, I'm actually going to start by asking a question to you. And given the changing of the clinical trial paradigm for approval of products that will be used globally, this really can be challenging, because we need to look at the global regulatory and approval infrastructure. From the WHO perspective, what are some of the untapped areas of cooperation and collaboration that you would like to see some action on?
Thank you. Yes, we're in difficult times, and difficult times because we've got good products. And certainly with trials, with future trials, I mean, the trials that were mentioned today, both had TDF FTC arms, and therefore one could anticipate, if the products are as effective as we hope, that one would be able to see superiority, because the products rely much less on adherence. But of course, you know, in the future, if one's comparing them with other long-acting products that don't have that adherence issue, but do have very high efficacy, numbers of events are going to be so tiny that one will definitely need other measures to look at, you know, the counterfactual, you know, the placebo arm counterfactual estimates. We would have concerns that using the recency assay as at the enrolment as the main feature to derive this counterfactual estimate would be risky, because I think the characteristics of people who test positive at enrolment would not necessarily be the same as those who test negative as they're entering, possibly coming into a PrEP trial. So triangulation with wide ranges of other data sources, as has been described, will be absolutely essential. And although, of course, background rates, and as Jessica Jetman has described, by using recency assays in surveillance, will be helpful to ascertain a background rate. And we also have similar ways of looking at that from modelling estimates, from the Naomi modelling, which goes down to district level, incidence amongst disaggregated by age and sex. Is also very helpful, but of course not directly relevant to people coming into a trial. So I really want to confirm what and agree with what many of the presenters have said. We need to be very collaborative and get all the data we possibly can, both from surveillance, both from modelling, both from other trials, as well as looking at the recency of people going into trials.
Great, thank you. I will let you then go straight into the panel discussion. Rachel,
Yes.
Thank you.
Thank you very much. We've got a great panel here. We've got I'm not quite certain whether it's 4 or 5. But we're going to start off with Connie Kellum, who's, I'm sure, well known to so many people. She's a professor of global health and medicine for allergy and infectious diseases at the University of Washington. She has huge experience. She's been involved in many PrEP prevention clinical trials, such as Partners PrEP, and she's a co-investigator of the Micropsides Trial Network. We have Aisha Kazami, who is a senior scientist and epidemiologist at Caprisa. Her experiences includes the involving HIV epidemic in South Africa and factors influencing HIV acquisition, especially amongst young women. She's the principal investigator for the HIV Incidence Provincial Surveillance System and the HIV Transmission Network Studies. Next, we have Grace Kumwenya, who is an HIV prevention advocate, currently working as chief of party at the Key Population Programme in Malawi. She's an AVAC advocacy fellow, passionate about women's health. And Kathleen McQueen is a senior social scientist in global health research at FHI 360. She's got extensive research experience with social, behavioural and ethical aspects of biomeasurable prevention trials, both domestically in the US and internationally. And she's also an adjunct associate professor at the University of North Carolina at Chapel Hill, as well as the Gillings School of Public Health. And finally, there are two panel members from the FDA, Peter Mayel who's a medical officer and reviewer at the Division of Antiretrovirals, and Tamban Valapil, who is at the statistical team in the same department. So I think I'm going to start with Connie Kellum. So Connie, you're an experienced clinical trialist who's navigated many international settings and contexts successfully. In an ideal world, how would we triangulate between the various sources of data to inform the counterfactual placebo estimate? And what, if anything, are we missing?
Thank you, Rachel. And this is a very timely discussion. I think that we don't have all the answers, but I guess in my ideal world, let me focus on trying to focus on strategies in African adolescent girls and young women. I think that we've heard about some of the potential tools. One that I'm hopeful for is that the data that will come out of the recency testing of young women presenting to outpatient settings and other settings that Jessica Justman talked about, the TRACE initiative, because I think that is probably least biased in terms of reflecting incidence in the same time period among women who are not selected based on their interest in and willingness to be in a clinical trial. So that would be one. I think secondly, we have relatively recent studies, both HIV vaccine and other non-vaccine prevention trials that give us an estimate of HIV incidence when PrEP use was quite low. So for an example, again, focusing on African adolescent girls, young women, that would include HVTN 702 and soon probably HVTN 705 and ECHO. And I think those, and for MSM in other settings, it'll be the Mosaico trial. So those will give us basically a placebo arm incidence in at least within several years of when new trials are being launched that will have relatively low PrEP uptake. So again, assuming that our eligibility criteria are similar to what we're talking about for the PURPOSE trials and EMPOWER trials, I think those will be very important. A third kind of contemporaneous estimate of incidence would be PrEP demonstration projects. And again, that's maybe benchmarking it at the other end where people are using some PrEP products with various levels of adherence, and that would give us probably a lower estimate of HIV incidence in people who know they're on something effective and are using it as best they can. But we know from multiple studies that oral daily FTC-TDF that falls off over time. So we'd get a sense of what that trajectory looks like. And as Dr. Das talked about, we can also use drug levels to try to estimate how well they were using it among seroconverters in a random sample. And then I think the other way we can do it is math. A fourth area would be math modeling to try to take risk scores like the voice risk score and try to estimate based on the characteristics of the population we do enroll what we would predict the incidence would be. I think we've learned we have to be conservative in our estimates there because we can overestimate incidence easily if we don't take into account temporal trends in testing and treatment and viral suppression, but that could be another measure. So I think those are the main ways. I personally, probably the last thing that we haven't really talked about is a contemporaneous cohort of people who are meet the eligibility criteria and who don't enroll, but I think you would get, it may be very hard to enroll a sufficiently large cohort to get a precise estimate of incidence. And some of the demonstration projects of PrEP, you get 90% uptake. So how much can you really learn from the 10% who don't want to enroll in a active comparator trial of a novel agent? I'm a little concerned it would be a lot of effort and still be subject to a fair amount of bias. So given everything else that trial sites and sponsors need to do, I'm not convinced yet that that's really the direction we should go.
So thank you
back
so
to you.
much, Connie. Yeah, those are very valuable thoughts. We need to really make sure we use all data sources to the best of what they can provide. Let me turn to Aisha. And, we talked a little bit about the recency assay and how that could contribute. And this is something that you have a lot of experience with. You've recently also published a comprehensive report on HIV and prevalence and incidence trends in KwaZulu-Natal. What are some of the key lessons that you have learned from working on this project that are relevant to this project when we talk about using the recency assay as a public health tool for the purposes of a PrEP trial?
Thank you, Rachel. So it's actually really exciting to see that the methodology is being sort of discussed and hoping to move this forward. So with the recency-derived estimates, HIV incidence estimates, specifically from population-based surveys, we've actually compared these to the direct estimates that we've through our cohort data as well. However, we found that the recency-derived estimates are innovative, they are useful, and they were very important as we could generate these in a short time period. And of course, they are useful as a public health tool for the rapid assessment of the impact of programs, and specifically to understand the status of our HIV epidemic, especially in settings such as rural KwaZulu-Natal, where I actually worked from as well. However, I also consider that when we apply these to PrEP trials, there are some caveats to deriving and applying these estimates. And for one thing, I feel that it's important in terms of the timing. For use of these estimates in PrEP trials, we actually need to make sure that they are generated closest to the time that they are likely to be applied. So in essence, I think this was already discussed, but it's clearly important to be applying these timelessly as well. The second thing that I want to just highlight is the importance of the sample sizes. And especially for the recency-derived estimates, we found that you need really large sample sizes. And so what would be the sample sizes to ensure that we have a precise measure with narrow enough confidence intervals so that we can reliably estimate these as well. The third thing I also considered was in terms of precision and validity. So we found that recency-derived estimates are importantly derived from the use of several parameters, and some of them being the false recency rate or the mean duration of recent infection as well. And so what would the FRR be at the time of undertaking these trials as we are rapidly scaling up ART through the universal test and treat strategy? So how will the FRR be in the context of the scale up as well? So I specifically consider FRR to be an important parameter, especially as we have an evolving epidemic and we have these evolving parameters through the implementation of and the scale up of ART as well. And especially we find that many of the countries are really ramping up in terms of ART provision to be able to achieve the targets that are being set by UNAIDS for 2025 and 2030. So clearly these recency assays derived estimates are important, but we need to consider these caveats because they are really evolving and they're evolving very rapidly. So we need to be upfront in terms of how we actually apply these estimates.
Right, that is so true. And thank you so much for your insight. Back to you, Rachel.
Yes, thank you, Aisha, for that cautionary note there. Turning to Grace. Grace, you've participated in many community consultations with the three current phase 3 programs sponsors and many other organisations. Now that these 2 programs are being rolled out, What will you be watching for with respect to community confidence in this approach?
Thank you so much, Rachel. As communities, one of the key things that we are watching out for is the implementation of good participatory practice guidelines. These documents have been available for quite some time, and they've guided how research or scientists should engage with communities. So what we're looking for is basically how sponsors and researchers who have a little bit more knowledge on how to engage communities. I think from the experiences that we've talked about today, we are seeing a lot of engagement happening right now, but we do want and expect a little bit of a more structured approach in the way that communities are being engaged. At the same time, we do look forward to resourced engagement, because this is a space where we want communities to be coming together to learn what is happening with this new approach. And we do need a little bit of more investment in this space. And when we talk about participation for communities, we also need to think about how we move from the global platform to the host countries where this research is being, and then move from there to even the sites themselves. How are they being engaged? For me personally, I feel like with the new design, with the new approach, stakeholder engagement will become even more critical, because there are some serious questions that this is raising for us as communities. Probably scientists, researchers have kind of been in the space for some time. They understand how all this works. But communities, as they're coming in, they'll raise questions. They'll have questions such as, how will efficacy be measured in this context, where we are talking of background incidents? They'll have questions like, how will listeners' results be handled, especially in countries where results are being communicated to participants? They'll have questions like, how will data that is being brought on the table from previous studies, how will that be, in terms of informed consent for previous participants, how will that be handled? So we do acknowledge that where we are right now has been a result of science and research, and that has been a very important milestone. But at the same time, as we get into this exciting phase of this new approach, what we are asking for is, let's get a little bit more structured. Let's have an education plan for communities so that we move together in this path. So I think for us as communities, that's what we're looking for. Whatever language is coming out, we want to be part of that conversation in a structured and more concrete approach. I think I'll leave it there, Rachel. Thank you.
Thank you very much, Grace, and a big plea for involvement of communities and support for educating communities on these new methods. I'm sure everyone is listening to you. To Kate McQueen, from a social scientist perspective, what are some of your thoughts regarding a primary efficacy analysis based on a counterfactual placebo estimate? What social behavioural drivers do we need to be aware of as we evaluate study outcomes?
Thank you, Rachel. And I want to start by saying, I think that what we've heard from everybody who's participated in this incredibly valuable forum is that there is an astute understanding of how important the social underpinnings and overpinnings of all of this work are. HIV, it's socially constructed. We know that. In the trials that we talked about today, each of the PrEP modalities is being investigated in 2 trials, and the 2 trials will differ by gender. They're also being implemented in diverse cultural, social, economic, and political settings, and we know that background HIV rates differ, and a lot of that has been touched on. I think it's really important that that become a systematic base of knowledge itself, and not a series of disparate insights from across all the different disciplines and concerns engaged, but that we really find a way to integrate the questions and integrate that with what do we need to ask, what information do we still lack, and what do we need to be careful about. We know from our experience with the early oral PrEP trials that if we observe different efficacy outcomes in trials in different populations with the same intervention modality, that this can have major implications for equitable drug access. You know, is something working or not working, or is it simply the way we measured it, or our unawareness of a basic bias that's socially driven?
And so in those trials, adherence was socially constructed in unexpected ways, and the result was that access to PrEP was delayed for women. So what I would throw out is that we need to really talk about, as a coherent set of issues, what are the social epidemiological research questions that we need to answer?
Right.
And as you know, with to end with, we need to keep a bit of humility and keep an open mind regarding what we don't know that could be driving the observed incidence outcomes in all the variety of ways that we're measuring incidence.
Thank you so much, Kate. And I apologize that we're running out of time. I will just thank everyone profusely again. Also Caroline and the IAS team for making this possible. Participants will have the opportunity to continue asking questions that you can submit, that will be emailed to us, and we will attempt to answer them as promptly and as quickly as we can. So thank you so much again. Wonderful discussion. Obviously, a lot more to talk about and resolve, but I think today we had the best team that could possibly be gathered to really start some of these in-depth discussions. So thank you very much to everyone. Have a wonderful rest of the conference.